Hi there. This is the first video in a series of 8 discussing genomes in detail, and this one's the introduction. So I'll just talk about genomes in general and then also introduce what the other 7 videos will contain. So here's a breakdown of the 8 videos. This first part is just a general introduction. Then part 2 will be on RNA viruses, where I'll go into quite a bit of detail. And then part 3 is DNA viruses, into a different type of detail. And then part 4 is on bacteria, the first part of bacteria. And part 5 is a continuation of the bacterial genomes, plus archaeal genomes. And then I'll talk about endosymbionts and parasites as we move towards organelles and eventually eukaryotes, which have a lot of endosymbionts that have become organelles, and still quite a few endosymbionts as well. So there's a lot of detail I can present on RNA viruses, so I'll give gene details, sequences, things like that. For DNA viruses, I'll talk about gene details plus expression of those genes, because several of the DNA viruses have been explored in detail, and then some controls. And then bacteria, I'll talk about gene categories and proportions, so not as much detail on the genes themselves. Archaea also gene categories, proportions, and comparisons. For symbionts and parasites, I'll talk about gene composition, but also reductions in genome, which often happens or almost always happens, as well as interactions with the host, and then into genome composition of organelles, sequences, reductions, interactions with the host cells, and then some of the chimeric biochemical pathways. And finally, with eukaryotes, since they have much larger genomes, I'll talk about genome composition and some on comparative genomics. I'll concentrate primarily on genomics, which is the genome is a complete DNA sequence of an organism. Transcriptome would be the complete RNA sequence of all transcripts, and that's pretty much looking at expression of the genes. And then the proteome is comparison of the protein sequences of an organism, again related to expression of those genes. And then the metabolome is the complete list of metabolic pathways and enzymes of an organism. As far as being able to determine each one of those, the genomes, you just sequence the entire DNA. Transcriptomes sequence the RNA. Proteome is a little more difficult in that you have to separate different types of proteins and then try to elaborate what each one of them is. You can actually take the transcriptome and make a proteome in your computer, and so there's a possibility that way. The metabolome is a little more difficult in that you have many different types of assays for each category of biochemical molecule. However, you can deduce a metabolome by looking at the genome and transcriptome and proteome and come out with very good guesses as to what those bacteria or organisms are capable of doing biochemically. So the total genome of an organism is the chromosomal DNA plus the mitochondrial DNA, chloroplast DNA, or other DNAs that the organism has within it. For an RNA or a DNA viruses, it's the RNA or DNA of those chromosomes. And you can talk about just the nuclear genome and the mitochondrial genome as well. So here's just a couple of examples of that. Bacteria have usually 1 or more chromosomes. Some have up to 5 chromosomes, plus sometimes plasmids. DNA viruses have 1 or more chromosomes made up of either single-stranded or double-stranded DNA. RNA viruses, the same way, they have 1 to several chromosomes made up of single-stranded or double-stranded RNA. And then for eukaryotes, you have nuclear DNA here. You can also, most of the time, have mitochondrial DNA, but not always. You can have chloroplast DNA, but again, not always. And then in the chrome alveolata, that is the Stromenopiles alveolata, some of the excavata, rhizaria, haptophytes, they will have the remnants of a eukaryote inside of them, which is an organelle. And so each one of those compartments can have DNA, including a leftover nucleus, mitochondrion, and chloroplast. Most often they'll have the chloroplast DNA, but then a lot of the DNA that was originally in the organism and the organelle has moved to the main nucleus of the cell. This is just a sample of some of the sequences that have been determined over the past several decades. So the MS2 RNA virus was the first thing sequenced, because at that time in the 70s it was easier to sequence RNA than it was to sequence DNA, but it took several years just to do that. So it has a 3.6 kilobase genome size, only has 4 genes, and that's a gene density of 1.18 genes per kilobase. Everything in white here, white font, those are all viruses. Anything in yellow, those are organelles, and the orange down there is a bacterium. So you can work your way up through FIX174 with 2.03 genes per kb, HIV, which has 9.7 kilobases of DNA, about 11 to 15 genes, 0.93, so the density is now going down. And then up to influenza, which actually has 8 chromosomes, and all of them together equals 15 kb, 11 genes, 0.79 genes per kb. I will be discussing some of these in more detail in the next video. So you can see in general, these are just ordered from small genome size to larger genome size. So you have a symbiotic bacterium. It has the smallest known bacterial genome, and it's a symbiont of a different organism. So it has a reduced genome and fairly concentrated genes, 1.13 genes per kb. And then you see the human mitochondrial genome at 16.5 kilobases, 37 genes, of which most of those are tRNA genes, so they're quite small. So you have a higher gene density, mainly because it consists primarily of small genes and then a few larger genes. So it's 2.18 genes per kilobase. This is the complete sequence of the RNA bacteriophage or virus MS2. Sequencing was completed in 1976, and although it is a short sequence, 3,568, I think, it took several years to complete the sequencing of this. Here is part of the human mitochondrial genome, so you can see that it's much longer than the viral sequence just presented. 16.5 kilobases of DNA, and this is a DNA molecule, double-stranded DNA molecule. In red there are the small subunit of the ribosomal RNA and the large subunit of the ribosomal RNA. And then other genes in there are things like for cytochrome oxidase and ATPase, etc. Here is the next section of that genome, going up to 12,000 something, 12 kilobases. And here's the final section going up to 16.5 something kilobases. And that's the entire mitochondrial genome that you have in your mitochondria as well. So I think by this you can see that we can't present entire genomes and ever hope to get through them. But remember, this is 16.5 kilobases of DNA in the mitochondria. But in your nuclear genome, you have approximately 3.2 billion base pairs of DNA. So that would be a lot of volumes of books to print that entire sequence. Going back to this diagram, and if we work our way up through Ebola virus there at 19 kilobases, and then this SARS-CoV-2, which is the disease organism for the COVID disease, COVID-19, has one of the larger genomes for an RNA virus. And then working up to an Epstein-Barr DNA virus, those are much, much larger for the DNA viruses. And that's one reason that I've already talked about, is that RNA is limited as far as how large a genome can become because of the higher mutation rate, whereas DNA genomes, DNA organisms can have much larger genomes because of the lower mutation rates and the repair mechanisms. So moving up further, this is the second part of this. You can see there are some symbiotic organisms with larger genomes, 500,000 base pairs, and then into an archaea with a 1.7 megabase, million base pair genome. So these are free living organisms, and the cutoff for free living organisms is right around 1 million base pairs. And that has 1,700, almost 1,800 genes, and a gene density of 1.05 genes per kilobase. And then a bacterium, one of the first ones that was sequenced was Haemophilus influenzae, as a fairly small genome for a bacterium, around the same number of genes as Methanococcus. And then the Cucumis mello, which is muskmelon, mitochondrion, it's quite large, but it only has 33 genes and a very low density of genes. Moving up further into more bacteria, E. coli, about 4.6 or 4.7 kilobases for a genome size. And the density of genes in E. coli is about 0.93. So you can see that for bacteria, the gene density is staying right around 0.9 to 1.0, sometimes a little lower for Mycobacterium, down to 0.5, but it's remaining fairly constant. But once you get to eukaryotes, Saccharomyces cerevisiae, Plasmodium falciparum, now they have larger genomes, but the number of genes doesn't increase as a proportion of that compared to bacteria. So now the gene densities are going down to 0.2 to 0.5. By the way, the color coding on this is that the orange are symbiotic bacteria, the white again are viruses, light blue are archaea, red are bacteria, and then the medium blue, those are eukaryotes. Working up into all eukaryotes, including fungi, nematodes, insects, plants, etc., and Homo sapiens is down there too. You see the genome sizes here are getting quite large, including the amoeba, which has a 670 gigabase, or billions of base pair genome, and about 16,000 genes. And you can also see that the gene density is going down, so they have a lot more DNA, but all of that DNA does not become genes, that is mRNA genes encoding proteins. But that doesn't mean that it's all junk, as it once was thought. A lot of those sequences are doing many, many different jobs, including structural parts of the chromosome and controlling expression of the mRNA genes as well as other genes. And most of the increase in genome size also is due to repetitive elements such as microsatellites, mini-satellites, transposons, introns, and things like that. So quite a few different things moving parts of the genome around, as well as controlling parts of the expression of different genes. This shows that graphically. So as genome size increases, gene density decreases. although the number of genes often does not go down. So it's not really indicative of a drop in genes. It's just more DNA with the same number of genes, or sometimes a little more and sometimes a little less as far as genes. So at the top there on the right side, that's Homo sapiens, Drosophila melanogaster, Arabidopsis thaliana, Cinerobditis elegans, etc., going down. So you can see as genome size does increase, the gene density, number of genes per base pair or kilobase, however you want to measure it, decreases in general. As I said earlier, this is not true for bacteria. Pretty much bacteria group in one location where the genes per kb range from about 0.8, a few are lower than that, up to about 1.1 or 1.2, no matter what the genome size is. But as you get out to eukaryotes, and this is just for fungi, you can see that relationship breaks down so that you can increase the genome size by quite a bit, and the density of genes generally goes down by quite a bit. Here's another graph to illustrate that genome size at the bottom, and then the amount of coding DNA. So the amount of DNA that is encoding mRNA genes or mRNAs. So you can see that bacteriophage, eukaryotic DNA viruses, prokaryotes, and to a certain extent unicellular nuclei, that is eukaryotes, about 100% or close to 100% of the genome is made up of mRNA genes. But as you go to animals, plants, and multicellular protists, then that breaks down so that in some cases you have 1% or less than 1% of the genome actually consisting of mRNA genes. And in humans, it's around 1.5% of the genome consists of mRNA genes. Here's a bit more detail on that. So if you look at bacteria and archaea, as the genome increases, all of that DNA that's being added consists of genes primarily. So it's pretty much a straight line or linear relationship between genome size and the number of genes. So getting into eukaryotes, if you look at the bottom, there's the bacteria. They would be on that straight dashed line, black dashed line, going up pretty much linearly as you increase the size of the genome. Most of that being added, those are genes. But then you get into the eukaryotes, and that begins to break down so that there's a broader and broader range of genomes for about the same number of genes. And so by the time you get to amphibians, there's a wide variation in the genome sizes, although the number of genes remains fairly constant. And then you go up into flowering plants where, again, the number of genes is fairly constant, especially in the angiosperms, but the genome sizes vary widely. For many years, people were trying to figure out what all of that extra DNA was. So a set of studies looked at whether the number of chromosomes was related to the genome size. And as you can see here, all you get is a big blob if you plot all those points, so that it's not related at all to the genome size. Chromosomes, you can have a large genome with a few chromosomes, or a large genome with thousands of chromosomes, and vice versa. Molecular methods have been able to sort this out a little bit so that only a small percentage of the human genome, 1.5% there, are protein encoding genes. About 45% of the genome is made up of transposable elements, lines, signs, LTR retrotransposons, and DNA transposons. Duplications, simple repeats, and other heterochromatin, as well as introns, make up a large proportion of the genome as well. So those have been the major increases in the animal line anyway. In plants, they often contain mainly transposable elements. 5% of the genome is made up of mRNA genes, but 84% of the maize genome consists of transposons in at least 11 superfamilies. And then you have microRNAs and then introns and other things making up the rest of that. So transposons have really increased in most of the multicellular organism genomes. And in maize, anyway, this is one of the chromosomes of maize. There's a non-random arrangement of the transposable elements, especially the LTR retrotransposons. They're located from the centromere out to about 80% of the length of those chromosomes, whereas the genes that are actually encoding mRNAs which are translated into proteins are way out towards the telomere ends of the chromosomes. As you might expect, the amount of DNA in a cell determines to some extent the length of time spent in the cell cycle, especially in the synthesis, the S phase of the cell cycle. And this shows that in plants, you can see there's pretty much a linear relationship between time spent in the cell cycle and the amount of DNA per genome. And this goes up both for eudicots and monocots, and so it's pretty well been established. Also, there's a relationship between the amount of DNA, time spent in the cell cycle, and latitude, geographical latitude, so that as you go towards the poles, normally you find plants with smaller and smaller genomes so that they can go through their cell cycle faster and so that they can go through their life cycle faster. This relationship holds true pretty much through the temperate regions, but by the time you get to the tropics, it breaks down so that there are large genome and small genome plants at the tropics. There's not as much selection on life cycle timing and cell cycle timing. So here's a graph of genome sizes that I'll be showing in several of these presentations, going from RNA viruses down at the bottom through DNA viruses and then organelles, bacteria, archaea, and then a wide variation in genome sizes in eukaryotes. Protozoans have a very wide variation because that includes, all the way from excavata up to the Stramenophile. So there's quite a few different groups of organisms represented. So it looks much larger than maybe it would if those were broken down. And then up through the algae, that is the plants, through gymnosperms, angiosperms, and then through the Opisthokonta, which is the fungi, the invertebrates, and the vertebrates. Each one of these little circles with a letter in it, those are some genomes that I'll be discussing in the next several presentations. Also on this diagram, all of the bars here that are white, those all indicate organisms that require other organisms for their survival. So the viruses are obligate parasites. The mitochondria and chloroplasts have to be inside of those cells. All of the black bars indicate organisms that are free-living, genome sizes of organisms that are free-living. And then in between there's sort of a gray bar. Those include organisms. Some of them are free-living and some of them are require other organisms for their survival. So here it's just summarized. The RNA and DNA viruses are obligate parasites. Chloroplasts and mitochondria require them to be inside those cells for their survival. And then in bacteria, you have some parasites, some symbionts, some endosymbionts in the white. The black bars indicate free-living bacteria and archaea, and then there's a gray area in between where some of those organisms are parasites and symbionts and others are free-living. And the same with the eukaryotes. You have parasitic plants, parasitic animals, things like that. You have free-living organisms, and then you have a mixture in the gray indicated by the gray bars. So from this, you can see that larger genomes are required for free-living organisms, and as organisms become parasitic or become organelles, they need less and less of that DNA in their genome, so those genomes tend to be reduced. And recall that DNA is moving around all the time, so you can get DNA exchanges between different organisms. You can have DNA moving from the organelles into the nucleus, and you can have endosymbiotic organisms having their DNA move into the nucleus of the organism where they're an endosymbiont. And this explains some of the changes in those genome sizes. So when you have independent organisms, they maintain their genomes because they need all of the genes in those genomes in order to survive. However, if you have a parasitic or commensal relationship, one of the organisms is getting part of its nutrients and requirements from the other organisms, so that the genes that normally would perform those functions are no longer needed. They mutate and then eventually are lost in the genome. So those genomes tend to shrink over time. When you have a mutualistic relationship, both organisms tend to lose parts of their genome, and that's for the same reason, in that they're getting some of the requirements from the other organism. So those genes that would normally perform those functions mutate and eventually are lost. And then in an endosymbiotic relationship or an organellar relationship, the genome of the endosymbiont or the organelle can be much reduced because they're getting most of their requirements filled by the host organism or the host nucleus. And so eventually they can lose 90% or more, and sometimes all of their genome. Here's a graphical representation of the same thing, so that this is all for bacteria and archaea. So you can see the free-living organisms up on the top right of the diagram, where the genome sizes are above about 1 million base pairs, and the number of protein encoding genes is around 1,500 to 2,000 and above. And then you move into the obligate parasites and endosymbionts, where you drop below a genome size of 1 million base pairs and drop below the 1,500 genes, protein encoding genes, going all the way down to very small genome sizes ranging from about 100 to 200 or 300,000 nucleotides, and having fewer than 500 protein encoding genes. And then you get down to organelles, where they're to the point where they're almost losing all of their genomes and all of the genes, down in the tens of genes for the most part. Another curious thing that happens as organisms evolve from free living to symbiotic or mutualist to pathogens, and then finally to organelles, is their GC and AT proportions change. So that in free-living organisms, they have usually a GC content of 50% and above. But as you go, as things evolve into pathogens or symbionts or endosymbionts, they lose the GC pairs and tend to be more AT rich, so that by the time you have an organelle, they're AT rich and GC poor. This is probably partly due to the fact that the genes are mutating, and you're having changes from the C to T changes occurring more often, because those genes aren't needed any longer, and then eventually blinking out or going extinct. So you have a general change to more AT pairs. So over the next few presentations, I'll start by talking about RNA viruses, HIV, influenza, Ebola, and coronavirus, and then the presentation after that will be on DNA viruses. And then I'll move up from there into bacteria, archaea, endosymbionts, parasites, organelles, and then finally eukaryotes. Okay, so that's it for this time. Next part, as I said, is on RNA viruses. So thanks very much. See you.